The title of the article was Ingested Plastic Transfers Hazardous Chemicals to Fish and Induces Hepatic Stress. The presenter's name is Dion Wooden. This is the introduction. The casual question is, what is the effect of different types of microplastics on the livers of fish? The hypothesis being tested is that microplastics will transfer the tissue of fish upon ingestion, specifically the liver tissue. The prediction is that after the plastic transfer, it will cause bioaccumulation and will cause health problems, specifically stress on the liver. The relevant background information is that plastic debris floats in oceans all around the world, most of which is microscopic. However, the hazards associated with the microplastic debris on the health of fish are largely unknown. Therefore, the research objective is to find out the adverse health effects of plastic ingestion by fish and find out how to go about assessing the effects and dealing with it with the considerations of the hazards of microplastics in mind. The test subjects were Japanese Medaka because they were widely accepted model fish species. They were exposed to a low-density polyethylene diet because it makes up the largest portion of global plastic production. The fish were separated in 3 different groups based on the diets pre-made for them and placed in 3 separate tanks with the same controls such as water temperature, water pH, and such. One group was the negative control group, which had no LDPE. Another group was the virgin plastic group with virgin pre-production plastic, and the final group was marine plastic treatment group with LDPE deployed deployed in Urban Bay. The different treatment groups of plastics were prepared prior to experimentation and dried into pellets to feed the Medaka fish. The specialized dyes were sprinkled into the respective tanks of Medaka over a 2-month period. After 2 months, the Medaka fish were killed and the livers were examined for certain chemical pollutants to see if the microplastics had indeed transferred to their livers and caused damage. The results showed that the controlled diet fish liver with no LDPE shows that it was glycogen rich, while the other 2 groups showed that they were glycogen depleted, the 2 groups being virgin plastic diet and the marine plastic treatment diet. As shown in the figure, the B and C picture shows that the diets with LDPE in it can cause adverse health effects on Medaka's livers, while the controlled diet showed that it was normal and healthy. The data does support the hypothesis and the predictions because there is evidence that does show that the microplastics transferred to the liver tissue of the Medaka fish and caused adverse health effects, while the diet that had no microplastic caused no adverse health effects on the Medaka fish. The experiment demonstrated that microplastic ingestion by fish can cause hepatic stress, which is stress on the livers. The implication of the research shows that it is time to implement more extensive research that can result in effective policy management, including the invention of materials that are sustainable and safe for people, the environment, and wildlife. Since plastic waste will reach 2.2 billion tons annually by 2025, and it will affect the health of many fish. And just like fish, bees need saving too. I contributed this video. The video is made in the context of this course, which is Bio 281. Conceptual Approach to Biology Majors 1. Arizona State University, Fall 2018. Finally, Jesus.